lecture. So that if you work in, at least in Europe or UK with animals, then you need a license to work with animals. So in different countries it's different. You always need a license, but the question is how strictly or strongly they force it. So in Estonia you need a license. To get the license you should pass the course, local course, 2 weeks. They are teaching you everything, how to touch the animals, how to kill the animals. And after that you have official paper, you can work it. Or PhD students working with animals, they are passing this course. Sometimes it's a less strict, so that if you do some animal experiments but your supervisor has a license, then you are considered as a helper only and you can do it. In Estonia it works, in UK maybe not. And the question is whether they accept licenses from outside. In Estonia they don't want to accept. If it's from EU then okay, but if it's from the third country then they don't care very much. I don't know about Germany and other countries, but if you have already the local license, it's anyway easier to pass the course in the country you are going to PhD or postdoc. Then another thing is that I told you that you usually start from simple model and then you will go to more complicated model. So this is what I am doing. But I know very good people doing it very differently. We have one cooperator, very high quality researcher from Taiwan. They have very good transgenic core facility and they are very experienced to make the transgens, to study them. They are doing it differently. If they have some compound which could be good, they have some target which is very relevant, interesting, they will first check it in vivo in mice. Is it really working there? And then they will go to study the mechanism. And it makes also point, because when I'm doing my research 5 years, I'm studying the mechanism and then I discover that there is nothing in vivo, then why I did it? So she's quickly checking, is it working in vivo? If it works, then she will spend 5 years to do in vitro experiments, and she can be sure that this is really important thing what she's studying. So that vice versa is also possible, not just as I told before. Okay, so let's go to drug development. So it's now very generic what I'm speaking, but I have a feeling that most of the researchers need that general knowledge as well. So it's about drug development pipeline, then that you always should know what will be at the end. Then what you do, you should understand what is the unmet clinical need if you develop something. Then it's about the target product profile and a little bit about the current developments. And then we have this seminar after that shortly. I will give you for groups some rare diseases, and you will have to develop or do this target product profile for these diseases. So the drug development pipeline is this classical tube that you have discovery phase, then preclinical phase, then in states you have this investigational new drug application to FDA, then you have the safety, clinical phase safety and clinical phase efficiency, and then after that you may get drug registration. So the first phase where we are working, this is discovery and development. We will need targets. We need targets which we can modify which will affect our disease. Then we need compounds to that target. We will do the testing, screening, whatever. Or it could be an opposite, vice versa as well, that we have some kind of drug candidate which is doing something interesting, but we don't know how it works. This is not enough. We should have its target, or we should at least know what it is doing, what is the molecular mechanism, what is the biological plausibility, if it's correct phrase, for that drug effect. If you have a just compound which is not working, which is working but you don't know what it is doing, then it's limitation already. For example, this is limitation with these plant-derived compounds, that it's very difficult to understand what exactly they are doing in this cell. You don't get this mechanism so easily, and this limits their relevance a little bit. If you have a compound, you have a mechanism, then you should be sure that is it possible to administer it? Is it absorbing? What are the interactions? And you can develop that further to make it better. Then, and what could be the possible side effects? Then you will have to do the testing. You should have some kind of tests, in vivo tests in animal, to show that this compound is protective. Then you should do the proper toxicology tests to show that it's not too dangerous, it's safe. And all these toxicology tests at the end, you should do following the good laboratory practice GLP, which is quite complicated. Usually this phase will be outsourced to specific company doing toxicology. And then in States at least you will submit this investigational new drug application, and you can do the clinical research after that. At the beginning phase 1 it's safety, small amount of people. Then phase 2 it's the efficacy, how it works in small amount of people, and phase 3 is a bigger test. You will see efficacy, safety also from there. So after this third phase you are able to register it. So that it's slightly more complicated. The phase 2 could be 2A and 2B, but okay. So, and if you have at the beginning 10,000 compounds, then in preclinical phase you have 250 compounds. Then in clinical phase you have 5 compounds. Then you are lucky if at the end you have 1 compound that will reach to approval. So there are 2 agencies we consider most important. One is the US Food and Drug Administration, and another is European Medicines Agency. So why they are for us most important? One thing is that the biggest markets are there. So if you have a new drug and you really hope that it is doing something and it is making money, then you should start from the biggest markets. You can do it also locally here, I think, because you have a big market, but in Estonia you don't have market, 1.3 million. For us it's Europe and US for start. So they are checking whether your data is good enough. So if you will submit all your information to these agencies, they will check, is it safe, is it effective, is the risk benefit positive, and is it making sense to have one more drug in the market. After that, you can go to manufacture, marketing, and selling. If something happens after that, the unexpected side effects are coming, then it will be called back from the market. This is not happening so frequently, but it is happening sometimes. So this is the pipeline. So usually what happens is that academy is at the beginning doing basic research. Then in discovery phase you already have academy and industry, and from preclinical, clinical phases you have mostly industry. So industry is usually interested only when they are sure that it is safe. They will take it over maybe from this place here. And then basic researchers, they usually don't want to do too much. They don't want to go to preclinical toxicology. They don't have money for that. They can just do the basic research, show the efficiency, publish it. So this from drug discovery to clinical development. So this part is called frequently the valley of death or translational gap, because nobody wants to do it. Academy, no money; industry, too big risk. So it's not how to say always like that. Big pharma companies, also small pharma companies, are doing quite a lot of basic research, or maybe not basic research, they are at least doing a lot of drug discovery. So it's of course just oncology drugs, they are summarized. It's not a good example, but it shows that pharma companies are even in discovery phase, pharma companies are more involved than academy. And then at the end, it is only long. So that academy is usually finishing here or here. Not because they don't want to have a drug in the market, but just because they have no resources for that. So, but if you are starting from here, you are trying to find out that you have some drug, then you should understand exactly anyway what happens at the end. Because why to develop some molecule, to spend a lot of time for optimization and so on, if you don't care what happens with it at the end? So that usually you publish and nothing more, but if you develop it further, then it makes sense to develop it further only if it has potential to be a drug. So you should understand that potential. So there are a lot of things you should know. You should very clearly know what is the unmet medical need. You should know what is the expected outcome, how efficient it is. You should know what is the possible side effects, what is the risk of side effects, and are these acceptable. Then what people are not thinking, it's delivery and dosing. You may have a very good peptide in cell culture, but how you will deliver this cell penetrating peptide if you have a patient? And then money is also relevant. If you have a very expensive therapy, then who is paying? If you have cheap therapy, and even if the expensive one is in the market, your cheap one is competitive. So what is the competition? and what is the total cost. So if you are developing something, you should understand that somebody later will have to invest to this project. You can sell it, your university will sell it, or whatever, but you should understand that there will be buyers somewhere. And it's how big is the market, expected market. If you have a Wolfram syndrome, we have it, and then it's small. If it's Alzheimer, it's big. But then it's regulatory path. If you have a disease which have already the drugs, then this regulatory path is long. If you have rare disease, orphan indication, rare disease, you will get your drug to market much faster. Or if there is no treatment, you can get expedited review, as we had in this case of ALS drug in last lectures. Okay, then you should understand exactly also what is the IP status, so that patents are key components in drug development. If you have a drug candidate, then you should patent that, or your university should patent that at some moment, because the patent says that this is your drugs and there is something to sell further. So if you don't have a possibility for IP protection, for whatever reason, it's not novel enough, your drug, or you just don't care about patenting, then it's and you publish it before, then it's absolutely clear that this drug will never reach to the clinic. So that if you publish before, or you will even speak about it in conference, or I will speak about it here and it goes to the internet, then I can't publish, after that I can't patent this anymore, because it is known. What is possible is, which is slightly tricky, but you can patent and publish together. And what is less tricky and absolutely safe is that you will publish year and a half after patenting. If you have to speak about your drug and its nature before you patent, then it's necessary to use confidentiality disclosure agreements. This is removing the risk that this goes to public. Not fully, but more or less. So what you need, your drug candidate, what it needs to be patentable. So you can patent it if it's clearly new compound, or at least it's novel compound. There is slight difference here. So something is absolutely new and something is a little bit less new. So then if it's not absolutely clear that this is, it looks like invention, it's not obvious. This is not concerning drug candidates so much. Then it should be useful also that you can patent something which could be useful to somebody at the end. Then there are something you can patent. You can't patent gene sequence or protein sequence which is in the cells. And then most important is that to patent something you need very good data to support that. You can't tell that it's, this drug is good against Alzheimer, but you should describe it in very great detail to have convincing data, like in publications, to show that this is really protective. You should explain how this drug works, how you get this drug, and so on. So it's not so clear when and where to patent. If you patent too early, you don't have enough data, then you may not get the patent, or you don't cover all aspects, and later somebody will develop it further and do the new patent. If you patent too late, then it's risk that there are so many researchers in the world that somebody is doing the same thing, and you are not able to patent anymore. Then it's a very tricky question where to patent. So, if you will have a new drug in Estonia, then you can patent, or in India, it's the same. Then you can patent in your home country. Then you can patent in some other countries, US, UK, Japan. And then you have such kind of additional places. European patent, unitary patent, and international patent. I will explain them because it's an important issue. And timeline of patenting. So priority filing is the time you submit your patent application to your first patent office, wherever it is. Then time starts to go, and you will get this 12-month period after what during what your discovery is, how to say, safe, and you can during that period to submit it to other patent agencies, patent offices, and this priority filing date is showing its legal timestamp, saying that from this date as of this date I invented this. So after that, theoretically, you can publish it everywhere, papers, conferences, web, with small risks. So, but this is only one country. If you do it only in India, then it's not covered in US or in Europe. How to cover it in every place? It's not so easy. In this figure, it's this PCT filing, or it could be one US or EU patent. So PCT filing is that this is the Patent Cooperation Treaty, international treaty covering most of the countries. You can see the blue here. So you will give application, you will file it there, and you can do it only within 12 months after your first patent application. So people may think that after that I will get a patent in all countries, but this is actually a mistake. You don't get anything. You will just get extension. You will get maybe 30 months to patent it in all other countries you want. So it costs money, it doesn't grant you any rights, but it gives you time, because otherwise you have only these 12 months to submit it to other places. But if you have this PCT application also, then you have altogether 30 months. In some countries slightly less, in some countries, in India I think it's 31 also. So you will buy time for your patent applications. You don't get the patent. Then what you really want, you want to patent in big markets. US, Europe, UK, Japan are actually preferable for us, in Estonia at least. So we can have a European patent, or in Europe, which covers all Europe, or we can have a unitary patent, which covers half of the Europe, but which is much easier to get. We have not to translate it to 20, 30 different language. We don't have to pay a lot of money to deal with local people. Unitary patent gives us this half protection. You will see the red, orange place here, but fast and cheaply. So, what it is costing? If we have only US patent application, then it's maybe 10,000 euros. If we will have a local and after that US patent, then it's maybe up to 15,000 euros. If we will have Estonia, this PCT and US, then we have it up to 25,000 euros. Or if we have just main markets, then it's 35,000 euros. So it's quite expensive. And if you are developing a drug further than is your direct academic interest, then you should think about that, who is paying this. So but after that is also the patent managing costs will come in. But first is the filing costs. That if you really want to protect it in all main markets, you need a lot of money, and usually university is not giving you this money. If you establish some kind of small company and university give rights to this company, then this company should have this money. So we don't have this money. Once we had this situation, we had something to patent. We made application only in US because this is the biggest market. If you have it protected there, then and it's somebody will be interested, then you have some time to protect it somewhere else as well. So it seems to be some kind of legal story, but you actually, if you will do something, you will make grant application, then you should know everything that. Because at least in our grant applications, in EU grant applications, even in Marie Curie fellowship grant applications, you have the section IP management. And you can't write to IP management, yeah, if I will have something, I will patent it, or our university will patent it, because this means nothing. Usually these applications expect more information, and to write more information to this application, you should understand the patenting logic, timelines and costs. If you will get this application submitted, then there will be examination of the patent, which means that it could take first round half a year, but several rounds could be several years. So you will have a reviewers, actually quite similarly to journals, except that usually you have one patent reviewer in US. Quite similarly to journals, they are checking, is your finding novel? They will check the literature. You have claims in patents. You claim this is doing that. They will check, has somebody shown that before? And if they will find that it was published before, then it's not novel. You don't have anything. So in many cases, the examination says that it's not novel and you don't get it. Even if you pay the money to the patent attorneys and the patent offices already, but you don't get your patent. You have just submitted application, which you can use in CV. It's good for CV anyway, if you have a patent application, but it's really has no commercial value. If you get this patent or company or university, then it could be 20 years of protection, or it could be 25 years if you could extend it. Your discovery is protected for 20 years. But it's also not really fully protected, because other people can invent something how to cross your patent. If you have a compound patented, they will add few groups and they will get new patent. So then you have nothing, but you will sell if there is another patent. Maybe the compound is even better what they have. If the pharma company wants to develop similar drug, they will modify your drug a little bit and it's their drug after that. So that in patent application you should try to cover your discovery so well that it is difficult for others to cross or pass your description. Okay. So then another thing, you should very well understand the clinical need. So you should know, do you have patients who need your drug? So if we are speaking, for example, Parkinson's disease, then you can have very different kind of drugs. The best ever drug is the drug which stops the progression of Parkinson's disease. Or it's also good to have this levodopa, which is blocking the symptoms of motor symptoms of Parkinson's disease. But then you have also other problems. Parkinson patients have other problems. One of the non-motor symptoms is the constipation. 70 to 80% of Parkinson patients have it. And it comes sometimes before the motor symptoms. And there is drug candidate currently in clinical phase, if I'm not mistaken, which is theoretically at least effective against this constipation in Parkinson patients. So that the drug could be also against of these minor symptoms, which is problem. And if 70 to 80% of patients, Parkinson patients, have it, then it's a huge market, and constipation is a real problem. People will start to take this drug if it's effective. So that you should have, if somebody asks you what is the clinical need against Parkinson disease, in case of Parkinson disease, it is not just this what levodopa is doing, and so it's simple things. It could be also such kind of very different minor thing, which is problem for patients. So you should understand it, what patients need. Then another thing which is clear to understand is that what is the disease burden. That it's clear that Alzheimer's disease, having 50 million patients worldwide, is a very big market. So if you take the US Medicare Medicaid costs, so it's here it's maybe in billions, 200 billions per year. It's a really clearly huge market, and it's everywhere. Where there is money, there is need, because it's disease is everywhere. But then if you take a rare disease, what we're studying, Wolfram syndrome, it's more or less 1 patient per million worldwide. If we are estimating how many patients we have, it's 10,000 patients at the moment worldwide. Disease is very nasty. It starts with diabetes, then it's the optical atrophy, hearing loss, neurological symptoms, and so the time people are having very low life quality is 10 to 20 or more years. To treat these patients is very expensive. It's difficult to say how expensive it is, but what I found from one orphanet journal paper, that in UK the lifetime cost of treatment of this patient, one patient with Wolfram syndrome, is 1.5 million USD. So, of course, worldwide, the treatment cost is not 1.5 million per patient, but if it would be, then it's 15 billions already. So that even the rare diseases have a market. So you can say that there is no sense to develop drugs here. There is market if disease treatment costs are at the moment already so high. Then side effects and efficiency. So Alzheimer drugs, at the beginning symptomatic drugs we covered, and then now we have the antibodies, which are disease modifying therapies. What is the difference between the symptomatic drugs and disease modifying therapies? So in case of Alzheimer, you can see the graph here. This is the cognitive performance. Normal aging, it goes slightly down. If it's untreated Alzheimer, then it declines, going to close to zero with time. The aim of disease modifying therapy, if you get the diagnosis at that point here, then the aim of disease modifying therapy is to stop progression. This is perfect. The symptomatic therapy, this means you will get some improvement, but progression goes on. You have some improvement only. So it's a very clear difference. Symptomatic therapy, disease modifying therapy. Disease modifying therapy is always preferred. Then another thing is efficiency. So this is the Leqembi Alzheimer antibody. So we have here, can't read it, but some kind of parameter. 0 means everything is okay, and here everything is not so okay. And you see that without antibody there is disease progression. Things are going worse. With antibody, things are not going so quickly worse. It's 27% difference. It's not too much. It's okay, but it's not too much. If you will have another antibody coming to the market, you are working with another antibody. If your new antibody will have also 27% protection or 10% protection, then nobody is interested. If it's less effective, why to use it? Well, there could be other reasons, but if it's as effective, then why to shift to another antibody, only if it's cheaper? But if your antibody has a 50% protection, efficiency much better, then it's perfect. People will forget this Leqembi. They will start to use your antibody. Then you have side effects to consider. Actually, this is very important. Big part of the efficient therapies don't go from preclinical to clinical phase because you have side effects. If you take the same Leqembi, then what is the problem? This Leqembi as well as other antibodies, they are leading to amyloid-related imaging abnormalities. It is ARIA with edema, ARIA-E, and ARIA with hemosiderin deposition, ARIA-H. As I told, usually not a problem, but sometimes life-threatening, serious intracerebral hemorrhage, for example. And it's visible in MRI. Okay, you can see in MRI, this is figure, it's not MRI image, just a figure illustration, but it's visible as it tells, it's imaging abnormality. So let's see, will we get the video also?
Anti-amyloid antibodies used for the treatment of
No, we don't get the video.
often associated with amyloid-related image or ARIA, such as edema and hemorrhage. While the exact mechanism behind this
So it's not good day today, so we will not get the voice. But okay, we will manage. So that this ARIA, so we will see it. ARIAs will have a different symptoms. Headache, visual disturbances, confusion, focal symptoms. And this was the initial problem with that. Or maybe you can help with it. It was working yesterday.
201 trial. Analysis of data from the core study. A modest incidence of approximately
it's coming out from computer, but it's
bi-weekly and monthly lecanemab dosing regimens, and an even lower incidence of symptomatic cases of less than 3%. Radiographically, this incidence was mild to moderate in severity, with higher occurrence of ARIA during the early stages of treatment. Anti-amyloid antibodies used for the treatment of Alzheimer's disease are often associated with amyloid-related imaging or ARIA, such as edema and hemorrhage.
I can use this microphone here.
ARIA can occur spontaneously in Alzheimer's. Anti-amyloid antibodies used for the treatment of Alzheimer's disease are often associated with amyloid-related imaging abnormalities, or ARIA, such as edema and hemorrhage. While the exact mechanism behind this pathogenesis remains unknown, ARIA can occur spontaneously in Alzheimer's disease and has been associated with cerebral amyloid angiopathy. Incidents of ARIA can often result in symptoms like headache, visual disturbances, confusion, and, infrequently, focal neurological symptoms or seizures. Given the known occurrence of this adverse event with anti-amyloid antibodies, understanding the ARIA profile of emerging new anti-amyloid therapies can help in better therapeutic management. In this study, researchers from the USA report the in-depth ARIA profile for lecanemab, a humanized immunoglobulin G1, IgG1 monoclonal antibody, which has demonstrated efficacy in treating early-stage Alzheimer's disease. The researchers used data for 2 dosing regimens from the core and open-label extension studies of the double-blind, placebo-controlled lecanemab study 201 trial. Analysis of data from the core study revealed a modest incidence of approximately 10% ARIA-E with both the biweekly and monthly lecanemab dosing regimens, and an even lower incidence of symptomatic cases of less than 3%. Radiographically, this incidence was mild to moderate in severity, with higher occurrence of ARIA during the early stages of treatment. Moreover, the exposure-response analysis revealed high incidence of ARIA-E in ApoE4 allele carriers, particularly among the homozygous carriers, and an increase in ARIA incidence with increasing dosage of lecanemab treatment. The analysis also revealed a relatively low incidence of ARIA-H in the core study, which was dose-independent, occurred more commonly in ApoE4 carriers, and without the incidence of any symptomatic cases. Further, consistent findings were observed in ARIA incidence among the OLE participants, with 1 case of symptomatic intracerebral hemorrhage greater than 1 centimeter. In summary, these findings establish the safety profile of biweekly lecanemab dosing regimen with...
Okay. So they made this study, and from this study they concluded that in normal traditional patients the probability prevalence specific side effect is not so big, 6% if I remember it correctly. But then in some subpopulations it is higher, so that these ApoE homozygote patients, they had higher risks. So if you remember, this ApoE was risk gene for Alzheimer. So they have a higher risk for serious ARIA also. And because of that, it is, how to say, said that this ApoE status should be checked before the antibody treatment. And if they have these ApoE mutations, then it should be seriously considered whether the antibody therapy will be used or not, and patients and doctors should discuss the risks. So this is how the information from clinical trials will go to the guidelines. And what this means, if you're developing new antibody, this means that if your antibody is working better than this one, it's good. But it's also, if your antibody will not have this ARIA risk, or even if your antibody doesn't have the ARIA risk in these ApoE homozygotes, then your antibody is already having market somewhere. You can sell it to these specific patients. So antibody sometimes, or another drug, might be even less efficient than previous drug, but if it is having less side effects, then the risk-benefit ratio for this subgroup, for example, is better. Then administration route. So it's easiest way, it's pills and injections. Okay, you have infusions and other things also, but it's usually pills versus injections. So tablets are better, usually, because people don't like injections, they don't like needles, they like pills. But if you use, for example, GLP-1 agonist, you know GLP-1 agonists, what they are doing, diabetes drugs, obesity drugs. So you can have, mostly people inject them once per week, maybe, semaglutide, if I remember it correctly. But you can have also the tablets. So people who don't want to inject, they can use the tablets. But the problem is that this tablet goes to the digestive system, absorption is not perfect, it's slower, and your effects are weaker, or they come later, they are weaker. Sometimes these semaglutide have some kind of strange side effects. Of GLP-1 injections, it is like that, but in general people prefer tablets if possible. Then for all new drugs you need to know the pharmacokinetics. PKPD is the abbreviation, very quickly, what the body is doing with your drug. Because if the half-life is 5 minutes, then you don't have a drug, or half-life is 1 minute, because you have to take it 24 times per day. It's not working. Half-life should be long enough. Distribution should be good enough. If it's your Alzheimer drug and it's not going to through the blood-brain barrier, then you don't have a drug usually. And then metabolism and excretion. So if your drug is having very long, it starts to accumulate to somewhere, it is maybe not good, it may have bad metabolites and so on. So a lot of different things you should know very quickly. Then there is price. So if we take the same antibody, currently Alzheimer antibody, then its list price year ago in US was 26,000 USD. And then it was calculated in Lancet medical journal that if all European patients will start to use this antibody, then it will cost to EU to 130 billion euros if the price will remain the same. And this is half of the money used for pharmaceuticals in EU. So it's also, this means that there is no hope to treat everybody with this drug in EU, and if somebody will come with drug which is twice or more cheaper, then it will take the market, or at least the price of this Leqembi will go down. So if we have a Parkinson disease, then what is the unmet clinical needs? What do you think? Do we have drugs available? Yes, we have drugs available. Do we have disease-modifying drugs available? No, we don't have. Are existing drugs effective enough? So it is so. Levodopa is at the beginning quite effective. Do the existing drugs have unacceptable side effects? Yes, you remember these dyskinesias on-off coming after long levodopa treatments. Are existing drugs hard to use in case of Parkinson disease? No, they are okay. I mean, levodopa is in tablets, no problem. So if you know everything about your disease listed here, then you can do the target product profile. This is important thing, target product profile for people developing drugs. This is the desired profile of your future drug aimed to treat disease. It has slightly different meanings. In companies, it is the planning tool that you will have to reach to this and this, you have to achieve these and these things, then your drug development is going well. In regulatory agencies, this means that in US, to get this permit to go to clinical trials, your drug should be good enough, better than as good or better than previous ones, and this TPP is showing that. And in context of funding agencies or public health, it's also relevant, because if you are having grant applications, you are applying to get money for new treatment. You should explain there very clearly why your treatment you are developing will be better than current treatments. So if the disease has already many drugs, and all drugs have some kind of limitation, and your drugs theoretically will not have, then it's a big thing already. If current drugs are perfect, then your drug will not help further. So it's a planning tool for everybody. And it's not just a one-time paper. This is paper you can change during your development process. Just examples. Few examples. One example is the pain related to diabetic neuropathy, and it comes from NIH, National Institute of Neurological Disorders and Stroke homepage. So that first thing is that what is your disease, what your drug is doing. You have here the ideal results and minimum acceptable results. The ideal results is that all patients will have no pain or have less pain, and minimum is that here it's the same, but minimum is that what this drug should minimally do to be better than current situation. Then you should have a patient population. So ideally it should treat all the patients in this subgroup. All, for example, diabetes patients. And then what is your minimum? Here it's same, but in real life the minimum could be that it is affecting at least part of the patient. Then what is the treatment? Here it's chronic in both cases, but again, ideal is that it's one treatment just once. So if you take a gene therapy, it's only once. If you have antibody therapy, it's chronic. Delivery, it's again same, but ideal is maybe the pill, less ideal is injection. Then how frequently? Less frequently is preferable. Then what is efficacy? Best ideal result is, of course, that most of the patients will have a relief. But what is the acceptable minimum? 1/3 of patients will have relief. Here it's 30%. And side effects. Ideally almost no side effects, and minimum is that how big they could be. And then what is the therapeutic modality? So this is such kind of official. Then I made myself for Parkinson. So that if we have indication, so the best or ideal is that we will have neuroprotection, disease-modifying therapy for all patients. Minimum is that we have symptomatic treatment. Ideally is that we have all patients, old and young. Minimum is that we have subgroup. Then it's efficacy. We have some kind of endpoint. So it's significant improvement of motor symptoms, maybe 100%, and then primary is just statistically relevant improvement. And safety, safe compared with current dopamine agonist treatments. Treatment route, oral once daily, oral best, not so best, twice daily. And the price, cost effectiveness. Optimum is cost effective every way, minimum is cost effective compared with current therapies. So then pipelines. Just frequently the pipelines for drug developments are how to say, craft like that. Parkinson, Alzheimer. And you see that in the middle we have a phase 3, which is the last clinical phase, then phase 2, and first safety phase, phase 1. And then we have sectors, what they are doing, cognitive symptoms, or neuropsychiatric symptoms, this sector, or disease targeting, disease modifying small molecular drugs, and disease targeted biologic drugs. And here we have colors. Colors show the mechanism. Affecting amyloid, for example, is red. So that these graphs, they are available for the major diseases. They are very, very good. So you can see from here what is in clinical development, what is in pipelines. You can see how far they have reached, what is the mechanism and what is the general symptomatic or disease modifying, or what kind of symptoms are modified. So they are very good. If you have a rare disease, then you don't find such kind of graphs. Then you are looking for something specific, because these graphs, they are not fresh, they are a little bit old. Then you should go to the clinical trials database. So it's not including everything, but it's the US clinical trial database, which is the best. So I was asking about the clinical studies related to Wolfram syndrome. I got 11 ones. So they are telling what is the drug, they are telling who is doing this study, and what is the situation. And sometimes if the study fails, then it's usually not written here, at least in cases I checked. You can find it from somewhere else what happened. But you will get understanding what is under development. Then there is one rule, fail fast rule in drug development, that you should not go beyond your academic interest. If you can't see the unmet need, you can see that your drug is better than others, then just publish it. So if it is not more effective or it has serious side effects, then no hope. Or if you have something which can't be delivered, or if you already can see that there is no hope to find the money for patenting, or you know that university is not giving any money for patenting, then again, why to go on? Just publish and stop it. But then it's a problem that you may have a very good compound, you publish it because you have no money, and your compound will be not developed further by anybody because it's published. This is very sad situation. A lot of candidates will not reach to the clinical trials because there is no money. And this is why, where we had it, this is what is, sorry, this is why this part is called this valley of death. The drugs will die here. So this has been acknowledged. Little bit by governments, they give the money for this translational phase, and even more from private sector. So there are many private sector, NS, Spark, a lot of such kind of private foundations who specifically, Fox Foundation, others specifically, UK Parkinson, specifically funding this step. And these foundations, they usually not take your discovery, they just give you money for that step. They want that if they consider your discovery relevant, they want that this will somehow reach to clinic. They don't care how exactly, but if their money helps, then they will fund it. But they are very strict. I mean, you should have really good discovery. So we have a next coming, I don't know, after lunch, the seminar. And for seminar, I have 11 diseases. I can add a few more if necessary. And I would like you to form a groups of, I think, how much people? Groups of 3. You will take one of disease and you will generate for this disease the target product profile according to this table. So this is just example. You will, how to say, remove this part from here what concerns diabetic neuropathy, but you will have to fill this table accordingly to this disease you will get. So now we have a lunchtime, or we will continue on the fair. So if you do this table, you can come and show this here to other up to 3 minutes explain what is table. So maybe sometime here 20 minutes and we can meet at yes, okay. Okay, yeah. Okay, that's good. Take a picture from this. Actually, you should take picture from that, but we should divide you into the groups, and you should pick the disease. Yeah. Otherwise, it will be the same disease.
So all students will pick. I mean, we make a group of 3, and each one of you, I mean, each group should pick a different disease. So what we can do is we'll keep put all the names of the participants in the group, and every group will select the disease. And please do it right now in on the WhatsApp itself. All right. So do the literature survey, whatever you can find. Make this table. After lunch, you will be talking about these rare diseases. What are their symptoms? What are the